Sebastian, maybe you'd like to present the speaker today. I'll have to do it tomorrow. Okay?
It's okay, but you know, you can't do it.
Okay? Okay, good afternoon, everyone. We have the pleasure today of hosting a prominent speaker in the field of Alzheimer's disease, Professor Bartel Brugger from Lausanne in Belgium. And I think that we are really privileged that he agreed to come here. And yesterday I felt very bad because I suddenly realized that we're bringing someone for 3 days and making him give 3 lectures. So I would complain if that happened to me. But he took it very well. So you are invited to ask questions as we go. Some of the background material I already taught, but the rest is new stuff by a top researcher, and I also look forward to it. Thank you for coming.
So, pleasure to be here. Beautiful sun. That's something which we don't get in Belgium regularly. Today I will talk about, well, you can see the title, some hardcore biochemistry. And I think it's important to understand the biochemical aspects of a disease, because that's really giving you the hardcore targets for development. Tomorrow in the talk, in the Heller talk, I will talk more about genotypes and phenotypes in different mice we have studied. And there I will also focus on therapeutic potential. And then I have another talk, I think on Thursday, where I will talk about Parkinson's disease. It will be a completely different talk. It will be more involving Drosophila genetics and functional work. The talk on Thursday will be completely unpublished material, because we are not working that long on Parkinson, but I will talk about 2 kinases there. And today will be more a kind of overview, but I will go to very recent work we just published. And my goal is to give you an insight in the background of what we are doing. So if you have questions, interrupt me at any moment, no problem. If you don't get to the end of the talk, it's not bad. You can always read further. And I also don't believe that you have to know everything to be a good scientist. I think it's a question you raise yourself, which is more important than the stuff which has been published already. So, I think this is a very nice illustration of what Alzheimer's disease does with your brain. So these are 2 age-matched persons, Alzheimer's disease, non-Alzheimer's disease. The question is, of course, what causes this decay of the brain? And I want to discuss that with you, so I will give you some introduction on tau and amyloid plaques. That could be overlapping with what you know already, but I hope to give it a different spin. Then I will go quite deeply in the most important proteins involved in the generation of the amyloid plaques, the pristinins. And I will really try to clarify for you what the mechanisms are of the genetic mutations causing Alzheimer's disease. And then in the last 2 parts, I will talk about another enzyme, the beta-secretase. First, again, as a drug target, we have made some monoclonal antibodies which allow us to inhibit this enzyme quite efficiently. And then, if time allows, a few words on regulation of this enzyme. So, this you probably know, and this is a brain of a patient with these famous amyloid plaques and the neuronal tangles. And I think one of the most important insights at this moment is that these lesions, these amyloid plaques and these tangles, are not something fixed in the brain, something as an end result. I think we know now that this pathological protein aggregates under one or another form can be transmitted throughout the brain, can spread throughout the brain. And this is from a review just published by Cleveland, where he showed that similar to prime proteins, I guess you know about prime proteins, also a beta alpha-synuclein, which is important in Parkinson's disease, and tau protein can move throughout, infect neurons from neurons to neurons. And one of the most intriguing studies is with A beta, where a year ago or 2 years ago, it was shown that if you inject extracts from mouse which have a kind of Alzheimer's disease and you inject them in the peritoneum, the amyloid spreads towards the brain and you induce an amyloidogenesis in the brain. So there are still big questions around this. This looks fascinating and intriguing and also scary, but for instance, this column is very important. For instance, for A-beta, it's very unclear whether this really is sufficient to decline the whole disease. For prions, there is no discussion. So what I would like to say is the difference between aggregation and neurodegeneration. So to start with the biochemistry of these disorders, because we know that it can spread, but we know what you basically do, you take an extract of a brain and you inject it in the mouse, in the brain or in the peritoneum, and then you get the disease, but that doesn't explain you what are the real culprits, what are the real molecules which are doing it. And so the biochemical analysis of these lesions is extremely important. And so the tangles consist of a small protein, this tau protein, which is a microtubule stabilizing protein, and for reasons which are not very well known, I will summarize that in a minute, this tau becomes detached in Alzheimer's disease, and then, again for unknown reasons, it clumps into this amyloid, this tau amyloid tangles, which precipitates into neurons. So one of the points I would like really to make is that, in my opinion, the tau lesions are downstream of the amyloid peptide, amyloid lesions. And the reasons are, first, the genetics of Alzheimer's disease. So all the genes which have been identified in Alzheimer's disease, presennin, amyloid precursor protein, even ApoE4, all point to abnormalities in A-beta metabolism. None has been found to affect directly tau pathology. Second, there have been tau mutations found which cause genetic forms of dementia, but in that case you get frontotemporal dementia, which is a tau-only pathology. So in the Alzheimer's disease you get A-beta and tau. In the frontotemporal dementia you get only tau, which tells me that it can cause disease, but it doesn't cause Alzheimer's disease. And then there are some more indirect experiments, I would say, in mouse and cell culture, which show that if you, for instance in mouse, if you inject A-beta assemblies, that you can induce in one or another way tangle pathology. But these experiments you have to take with necessary caution, because these mice have usually already the tendency to form tangles, to form tau. They are genetically manipulated to get that type of disease. And so the controls on this type of experiments are not always very clear. Does this mean that tau is not important? And that's really a discussion which I think is now a very old discussion. What is most important, tangles of tau? I think that both are important. So first of all, tau is much more correlated with the spreading of the disease, with the dementia of the disease. And Braak, you have probably heard about the Braak stages of Alzheimer's disease. They reflect, in fact, the spreading of tau pathology gradually over the brain, correlating with increasing dementia. So the genetics of frontotemporal dementia, where it's clear that the tau mutation is sufficient to cause dementia. And then finally, also experiments again in mouse, which have shown that if you have an amyloid pathology, which sometimes causes some memory deficits in mouse, and you combine that with the tau knockout, you get an improvement of this pathological phenotype. So although tau is downstream of A-beta, it's still an important effector in the pathogenesis. But the big question with tau is that what makes tau toxic? So tau is normally present. What makes tau changing into something which causes neurodegeneration? And again, I'm going to summarize this because I basically think, I'm not a tau biologist, but I've read a lot about it, and I think basically nobody knows very well what drives tau from being bound to the tubule into these pretangles and paired helical filaments. So there is of course known that tau becomes heavily phosphorylated, but discussion there is whether this causes the detachment and aggregation, or whether the phosphorylation keeps your tau in solution and is a kind of protective reaction. There are also a lot of other post-translational modifications which have been implied in one or another way, glycosylation and others. Mutations can certainly cause and initiate this cascade. We know that from the frontotemporal dementia. And then finally, and that's something which I would like to stress, although I don't think that the evidence is conclusive, proteolysis could also affect tau and drive tau into a more aggregation-prone situation. I think it's something which the tau field under stresses. But if you look here to tau, maybe just to refresh your mind, tau is this quite unstructured protein, and it has 3 exons, exon 2, exon 3, and here in this repeat region there are 4 repeats here, protein repeats, and there is one which is also alternatively spliced. So you have 3 exons which are in and out, and so you have 6 forms of tau. And changes, especially in this splicing, have been associated with some tauopathy. So this domain seems to be important in tau pathogenesis. And so what in normal solution, tau tends to take this hairpin conformation. So it actually, well, with some goodwill, you would say it protects, or it makes this area, which is the crucial area for the aggregation in tau, it protects it and it solubilizes it. And we know, and again, I don't think that you have to remember all this protease. So there are a couple of caspases, there are some calpains, which have been shown to be able to cleave tau. And in general, in summary, what these different proteases are able to do is to cut away the C-terminus or parts of the N-terminus, and so basically releasing this center part, which is important for aggregation. And it's very interesting because in vitro you can drive tau aggregation by using fragments of this area. So what I think is that this proteolytic processing of tau could be very important to initiate the disorder. And if you go into the literature, there are only a few studies really analyzing this issue. Anyhow, so what I said already is that what the real problem with tau in Alzheimer's disease is unknown. So what is exactly the toxic species? What is the role of phosphorylation? Does proteolysis play a role in generating the toxic tau? And so the debate is basically, what do we need to do with tau in Alzheimer's disease? Do we need to restore its function? Do we need to drive it from a soluble fraction or an aggregated fraction into a form which goes back to the tau and stabilizes the tau proteins? Or do we need to take tau away from the equation because tau is in toxic species, it's solubilized, it affects membranes, or and the main argument there for taking it away is that if you have a tau knockout mouse, as I mentioned, and you cross it with an amyloid mouse, you improve the phenotypes. But again, I would like to say we have to stress that that this type of data are published in high-profile journals, but there are a lot of reasons to doubt that things will be as easy as that. One of the reasons is that the amyloid mouse which we use are very huge, highly overexpressing mouse, and you drive in a certain disease which is not necessarily the same as happens in Alzheimer's disease. So basically, there are a few mouse studies which suggest that taking tau away would be good, but I would like to see in vivo experiments in humans before I believe it yet. Sorry.
Yeah, I want to ask you, when you look at the tangles, and you have a wild-type forms of tau and mutated forms of tau, or only the mutated is the...
Both.
You have both.
And so also in mouse, if you have a mutated tau, you will also see that mouse tau starts to
And if
aggregate.
you overexpress, because it would be very easy to test whether it's a loss of function of tau, which is driving, or it's the tangle itself. If you now overexpress more of the wild-type, you make it worse or make it better.
I don't think that type of question is known. That's something you could do in Drosophila actually.
Yeah, I was
more
thinking
easy
so
than,
easy to
yeah.
do it.
The problem in Drosophila is that models used for tau pathology are these degenerating eye phenotypes, and there also again it's the same problem I have with the mouse, and maybe even worse. I collaborate a lot with Drosophila groups, so I start to understand what happens. So if you drive this tau to this high extent in the eye, then you get a protein disorder, but I don't know whether this is really reflecting the Alzheimer's disease. So I think that's the problem. So anyhow, so for tau, that's also the reason why I will talk mostly about amyloid, because there we have really experiments from human beings which show that amyloid beta is involved in the disease. And here you see here a very complicated picture, and you see the amyloid precursor protein, which is a type 1 integral membrane protein. And you see here this red bar, that's the amyloid peptide, which precipitates in the amyloid plaques and causes the formation of these amyloid plaques. And so the mutations which have been found, there are now, I think, 20 or even more different mutations in the amyloid precursor protein which cause Alzheimer's disease. And the remarkable thing is that all these mutations are clustered, are missense mutations, and they're all clustered around this A-beta sequence. So you have mutations here at the beginning of the peptide, in the middle of the peptide, and at the carboxy terminus of the peptide. And here you see a summary of all these mutations. Again, it's not important that you know all of them, but you see it's really remarkable that these mutations cluster around this site. And that's for me also very strong evidence that there must be something problematic with the A-beta sequence. So if you read the literature again, you will see that people claim that it's the intracellular domain of ATP which causes the disease, or that it's an insufficient, a loss of function of amyloid precursor protein which causes Alzheimer's disease. But I think that this data, which are human data, tells us something, and tells us, in fact, that it's the A-beta peptide which is problematic. So what are these mutations doing? Again, I'm not going into the details. Basically, summary: the mutations at this site, the Swedish mutation, they increase A-beta production because they promote the cleavage at this site. The mutations at the carboxy terminus affect, and I will go in much more detail later, the exact cutting of the amyloid precursor protein, and they provide longer A-beta peptides. And then finally, the mutations here in the sequence itself affect the biophysical properties of the peptide and make the peptide more prone to aggregation. So, and that tells us also something. Since these mutations do not necessarily affect the concentration of the peptide, the amount of A-beta peptide formed, but change the biophysical properties of the peptide, that tells to me that it's the biophysical properties of these amyloid peptides which are important. So this evidence, and Dennis Selku and John Hardy and several others became famous with this hypothesis. This evidence basically is the basis for the amyloid hypothesis for Alzheimer's disease. Well, I don't have to explain it properly. So you have the A-beta peptide, and it's an abnormal production of this A-beta peptide. And again, there is a huge amount of papers on what happens between the production of this peptide, amyloid peptide, and the final formation of this amyloid fibrils in the plaques. There's a huge literature about everything which is between. But this huge literature is not a consensus literature, and tells you, basically, if you go into that literature, that we don't know very well what happens there. But the problem is that if you do in vitro experiments, we have done them too, that pure A-beta peptide on its own, added to neurons or to cells, doesn't do much. And also the fibrils, if you mature this purified peptide to a full A-beta fibril, and you add it to a culture, it doesn't do much. So toxicity, and if you incubate A-beta peptide for a while, and you do some abracadabra, some magic, it changes, and you have a temporary window where these A-beta peptides are neurotoxic. And so that's the reason why we have at this moment this huge literature about toxic oligomers, A-beta star 56, ADLLs. Every lab has its own cooking procedure and ends up with something, but nobody knows whether it's different from the other groups, etc. So if you want to read more about it. So what I think is true is that there is a conformation here which is toxic. It's not unlike with prion disease, where we also know that there is some prion pathological structure of prion, but that's also not very well defined at this moment. So I think it's a bit the same with the A-beta peptide, but all the other claims, I would take them with a lot of critical attitude. And so we wrote very recently, and I don't think that I gained a lot of friends with writing this as a title, but this diagram basically summarizes 300 or 400 papers in the field, where you see all the different names given for all these different assemblies, together with cooking procedures, and we dare to put everywhere arrows because it's not a non-covalent change of this A-beta peptide. And so, of course, if you change the solution, if you change the salt, if you change the pH, it will change the structure of these peptides. So, if I talk about A-beta toxicity, I talk about a toxic A-beta soup. and let's see what the real structural biologists will find in the next decade or so, when they are able to really define what is a toxic species and what not. So this is the way I see the amyloid cascade. So you have abnormal production of A beta, changing either its biophysical properties or making it longer, or changing the overall equilibrium between the different A beta peptides. This causes an aggregate stress, which I think is a much better word than saying a specific oligomer is causing the disease. This causes tau. I've explained to you why I think that tau is downstream of this aggregate stress. And finally, and that's I think the big issue now, what is the link between aggregate stress, tau, and what is the link between aggregate stress, tau, and neuronal dysfunction? And there also we don't know very well what happens. We can discuss about this. There are a couple of hypotheses, but I think it's also again fair to say that we, for instance, don't know what the receptor is for this A beta toxic species. Although you will see papers published in high-profile journals. The problem is that there are several of those, so there's no consensus. And you will also see that people claim that A beta makes holes in the membrane. Actually, that's one of my favorite ideas. But again, they have then to use high concentrations. But there are others who say that it affects specific receptors, that it affects neurotransmission, that it induces apoptosis, etc., etc. I think that it's fair to say that we need much more research to understand that.
Sorry.
Yes?
Because there is no, there was, at least it was my understanding, probably wrong, that in some of the animal models, you don't have big aggregates, and that was one of the claims why people started to look into the oligomers. Or
I think the main issue is that you have a lot of discrepancies if you look to amyloid plaques and neuronal dysfunction. So in a mouse, you have a lot of amyloid plaques and very little neuronal dysfunction. But you have it also in human beings, and the Taoists, the people who said Tao is important, claim that already for 3 decades, that there are patients or people with a lot of amyloids in their brain, amyloid plaques, and which die quite normally. So there is a discrepancy there. I would like to be, that's why also why I think that we have to discriminate between amyloid aggregation, visible fibril formation, and A beta aggregation stress, which probably involves unstable species. And I think both show that there's something wrong with APP processing, but the overlap is not complete. And the basis for this is, I think we should give credit to Klein's group who published in '99 this HDLs. Nobody believed him, but he made one of these cooking procedures. He showed that there were no amyloid fibrils. He added this mixture to neurons, and he could show neurodegeneration. So that was in fact the beginning of the toxic oligomer hypothesis. And then a couple of years later, I think, so actually it has taken a long time before it was taken up by other researchers, but then it's published somewhere in Penicillin in 2001 or so, something about toxic oligomers. And then the field has exploded with a lot of confusing literature, by the way. Okay, so I'm not a biochemist, so I'm an MD, and we are used in medical sciences to work with black boxes. And you have a patient, has symptoms, you try to find out a little bit what happens, you give some medication. Does it work? Well, you have a good guess. Didn't it work? Well, either there is no good medication or you were wrong. That's the way medical science moves forward in simplistic terms. But but being involved in this Alzheimer work and being exposed also to good biochemists, I have the feeling sometimes that this is really the hardcore of the Alzheimer research at this moment. It has really yielded us insight in physiological and pathophysiological relevant processes. And so I think what we know is basically a couple of mechanisms how this A beta soup is generated. And I also think that these are really the targets, and if you can avoid the formation of this soup, I think that's probably the most rational way for therapy. So again, here you see this famous A beta sequence in the amyloid precursor protein, magnified. Important is that this A beta is sitting on the border of the membrane and the extracellular site, and that there are 3 proteases which can cleave in the sequence. So there is a beta secretase which cleaves here, quite straightforward, this enzyme. I will talk about it at the end of my seminar. And then you have the alpha secretase, which cleaves here in the middle. And again, this is the most abundant enzyme, most frequent cleavage. So if you look in liver cells or in skin cells or in any other cell type, apart from neurons, 90, maybe 100% of the APP is processed by the alpha secretase. And alpha secretase is mostly ADAM10 metalloprotease, also ADAM17, and maybe some other protease can contribute to this activity. In the brain, I think it's fair to say that about 50% of the amyloid precursor protein in neurons goes through the beta secretase pathway. So there is still a portion of the APP going to the alpha secretase pathway. And the field thinks that the beta secretase pathway, which gives you the first cleavage and then followed by the gamma secretase cleavage, gives you the full length A beta peptide. The field thinks that that's the pathological pathway, and that if you can stimulate this type of cleavage, that you will lower the load of A beta in the brain or somewhere else. I'm not so sure that this is really proven again. But to make it not too complicated, let's for the time being saying that alpha secretase is fine, that beta secretase initiates the amyloid cascade. And then the most important and most interesting enzyme is this gamma secretase, which cleaves here at all kinds of places in this transmembrane domain. And so I think people don't realize that, but maybe I have a citation from the paper. No. So when it was realized in 1987 by Conrad Bayreuther and a couple of his colleagues, that the A beta peptide, as they isolated it from brain, ended somewhere in a transmembrane domain of a precursor protein, they claim this is really not seen. It has never been seen that you can have a cleavage in a hydrophobic environment, in a membrane of a precursor protein. And they basically said, look, in Alzheimer's disease there must be something wrong with the membranes so that this protein can be released. And it has been really for I think it's fair to say 8 years, well more, 8 to 10 years before the biochemists started to take it seriously that it was possible to cleave here in the membrane. And the reason is that the biochemists started to believe it is that enzymes are identified who are able to cleave in this transmembrane domain. I think in our field, one of the most crucial findings in that regard was the identification of a gene, a presenilin gene, by Pete Sintrose-Wissop and later by a couple of other groups, who found that mutations in the gene encoding for presenilin were able to cause familial Alzheimer's disease, so an early onset familial Alzheimer's disease. And so when this protein was identified, and if you read the paper from Peter, it was absolutely unclear what this protein could be responsible for. So if you read the papers there, it was claimed that it had something to do with tau, or maybe with the membrane, or maybe with APP, but it was absolutely unclear. And what was also very intriguing at the time was that you see all these measles here, these yellow dots. These are places in the protein where missense mutations have been identified. And I should update it because in the meantime, I think 1/3 of presenilin has been found to contain 150 mutations have been identified. Different mutations have been identified in presenilin, all causing Alzheimer's disease. And so the question was, first, what is this protein doing? And two, how can all these different mutations cause Alzheimer's disease in a dominant inherited fashion? And this has really started the debate for at least 10 years of intense research. So I think one of the crucial elements in understanding presenilin was very basic research. So many of the research in the Alzheimer's field always want to have clinical relevant clinical relevant research. And for instance, they say presenilin is a mutation in presenilin are dominant. So what you need to do is to overexpress the protein because it's a gain of function type of disease. And so loss of function approaches will not teach you a lot about what's going on. My message to you is don't listen to that type of reasoning. The important thing to understand something is to understand the biological function. And the biological function you will never understand by overexpression studies. Because because that creates artifacts by definition. Loss of function and then restore the loss of function. That's the golden standard to understand function. And so that's the reason why we worked on the knockout of the precedence. And I think, and I'm still proud on that work, we could show that in the knockout of precedence that basically this amyloid peptide was not longer generated. And so they told us immediately that precedent was involved, was in the heart of the processing of the amyloid peptide. And it linked basically the protease, or a new gene, the precedent gene, to the already known amyloid precursor protein gene. So you have two independent genes pointing both to the amyloid cascade. And I think that was really, in my opinion, this has really changed my hesitation on the amyloid hypothesis to a belief. Well, you shouldn't use the word belief in research, but to a conviction that that's the right hypothesis. So, what I also would like to point out is that knocking out gamma secretase not only changes the A-beta 40, which is the most abundant A-beta peptide, but a whole bunch of other A-beta peptides, and I will come back to that. So if you go into the literature, you hear about amyloid peptides, but if you isolate A-beta peptides from brain, or if you do cell culture experiments, or if you do in vitro experiments, you will see that without doing anything special, you find several A-beta peptides. And you can see that here a whole series of these peptides, starting with 42 and longer, 42, 40, 38, 37 species even, they are all formed by gamma secretase. So gamma secretase is really responsible for the creation of a heterogeneous mix of A-beta peptides. So the second important point which the knockout experiments teach us was that precedent, well, genes are never made to cause disease. They have normally also a physiological function. And so an important thing we learned also from the knockout experiments was that precedent was necessary for normal notch signaling. And I have here an example. If you have a precedent knockout, you get this here you see nice vasculogenesis in a developing embryo, a mouse embryo, and here you see that this vasculogenesis is completely screwed, if I may use that word, in the embryos which have no precedent genes. And also here you see, for instance, this is the normal formation of an embryo with somites, and you see that these embryos, when they get, well, when you look at them in time, around day 10 of embryogenesis, that they are collapsed, and that's because the somites are not normally formed. And so all these things pointed to notch signaling problems in the precedent knockout mouse, and you can repeat that in Drosophila and all other species. So if you take away a precedent, you don't get Alzheimer's disease, but you get these embryological phenotypes. And just for your, I don't know how much you know about notch, but it's a fascinating research field. So in fly there's only one notch, but in mammalian there are 4 large notch receptors which are involved in all possible steps of embryogenesis. They interact with 5 different ligands, delta and JAK, and when they bind to each other, and that was not known at the time when the precedents were studied, but when they bind, when they engage to the notch receptors, you get the first cleavage in the ectodomain, because you get a conformational change of this ectodomain by an Adam-10. And then a second cleavage in the membrane, again, which then releases this notch intracellular domain, which goes then to the nucleus and affects gene transcription. And so one of the experiments we did was in the precedent knockout mouse, look whether notch signaling was disturbed, because of course it's not, when you have a phenotype which is similar to another phenotype, that you can automatically conclude that they are in the same pathway. But here you can really see that precedent is essential for notch signaling. Because here, for instance, you see the wild type precedent, and you see here the expression of delta, one of the targets of notch signaling, and you see here rather low signal. Here in the knockout you see a very strong in situ hybridization reaction. And the reverse holds true for HES, which is a typical notch target. So in the PS+, in the presence of precedent, you get a strong signal, and in the knockout, the signal is basically gone. So basically saying that in this, there is no notch signaling, because there is a positive loop there, and in the negative loop, where you have a negative feedback via the notch signaling, you get an overactivation. So that showed basically that indeed the notch signaling pathway was disturbed. And so then the question was how this happens, and again I'm going to summarize. So for APP we know already that you have first a cleavage in the ectodomain by beta secretase or alpha secretase, and then you get this carboxyterminal stub, and that gets cleaved by gamma secretase. So giving rise to the A beta peptide, right? That I have explained already. And so for the notch, you get a very similar phenomenon. So you get the ligand binding, you get then a cleavage here by ADAM10, which releases this ectodomain. You get this transmembrane stub, carboxyterminal fragment, which becomes then a substrate for presenilin, and which releases the notch intracellular domain. And this notch intracellular domain is then responsible, binds with transcription factors, regulates transcription factors, is responsible for the phenotypes I just discussed with you. So presenilin is really here in the, well, a second important point is once this was realized, the question was then what this ATP intracellular domain does, because of course you get the beta peptide, but you get also the cytoplasmic part of APP. Again, if you go to the literature, you will see that people claim that ACD is involved in all kinds of gene regulation. We did a lot of studies on it. I basically don't think that this is the case. When you look to ACD, the APP intracellular domain is 40 amino acids. Notch intracellular domain is 60 kilodalton protein, so it's a completely different game. And well, I'm not going to the discussion here. If you want more, we can discuss this now or later. But what you learn here also is that if you block presenilin, gamma secretase, you will block indeed A beta production, but you will block also this pathway. And this has really been the curse of the gamma secretase inhibitor experiments which have been developed by companies, etc. They have always been blocked by this notch toxicity. So another important point I would like to make here is that these findings came together with other findings in other fields. Maybe I should show this first. So findings in other fields which showed that presenilin was not the only intramembrane cleaving protease. So you have also the Cy2 protease family, which are metalloproteases. You have the rhomboid family, very, very interesting family of enzymes. And they are also enzymes with multiple transmembrane domains and with their catalytic sites buried in the membrane. And in the meantime, we have crystal structures of rhomboids and also of the S2P. And you see that this catalytic site is in a pit in the membrane. So the proteins around, the protein domains around the catalytic site form a pit in the membrane, and the substrates can go then from laterally into this pit and go into these catalytic sites. So it's really true that we can have hydrolysis in the membranes, and the structures are there. And the second point, which I think is important, this is in fact a complete novel way of signaling. So if you have phosphorylation cascades or other covalently modifying cascades of signaling, you have a kind of reversibility. So you can add the phosphate, you can take it off again, and you get something regulated. This is a very special way of regulation. So you have a signal, you get a proteolytic activity, you get an intracellular domain which is generated, and you get a genetic program. You can never go back. So this is Janus' head of Roman culture, where you look in two directions. Once this cleavage has happened, you have an irreversible decision in development or in any process which is regulated by this type of intracellular membrane cleaving signal. So, but let's go back to, and I promise you get some pause in 5 minutes or so, because it's a lot of hardcore biochemistry. But let's go back to our Alzheimer's disease and the presenilins. So until now I talked about presenilin as gamma secretase, but nothing is less true. Presenilin doesn't work on its own. And again, I'm going to show you the beautiful genetic experiments which have led to the identification of the 2 or 3 other subunits of gamma secretase. And I'm just going to summarize all these findings in this cartoon, which shows you the gamma secretase. So it's an impressive complex of 4 proteins with 18 transmembrane hydrophobic domains, nicastrin, APH1, PEN2, and then the crystalline with its catalytic site buried in this hydrophobic cord. Here you see, well, one of the big challenges for the field is to understand how these four proteins work together, and a couple of groups, including ours, are trying to purify sufficient amounts of pure gamma secretase to do crystallization studies and EM studies to get a full insight in the structure. Actually, an Israeli postdoc, Nadav Eilat in my lab, has recently also generated very nice EM pictures of purified gamma secretase. But here you see one of the published examples. The resolution at this moment is nowhere. So what we know is that under the electron microscope you have this big ball, which is gamma secretase, which is sitting in probably in a hydrophobic. This is probably the part which is in the membrane, and then you have some holes in the structure, but we don't know what they are serving for. So it's a bit frustrating at this moment, but this is the best picture we have of gamma secretase at this moment. That also implies that using structure function analysis to develop good drugs is kind of hampered. And I'm going to skip this because this is really very specialized. In a nutshell, what we did is we scanned the whole protein with cysteine, so bringing in cysteine mutations, and that allows you to find out neutrophobic and hydrophobic patches. We have published a couple of papers on that, but I will spare you that for because it's really that would take me a day to explain to you. So, do you want to stop here for 5 or 10 minutes, or shall we go first to this part and then give a pause? This takes me about 15 minutes of hardcore biochemistry.
He thinks that they need to drink something.
I think so too. Okay.
Okay.
I see already a lot of tired faces. Let's take 5 or 10 minutes.
Can you
make it? Good.
Wednesday morning. Next Wednesday.
So that means that you and I and Beula need to sit this way. Perfect.
Yeah.
Okay, so I'll say no that the team.
If check the time. In order for it to check, I believe the time.
Okay.
Maybe like 1 hour after they
Yeah,
get to.
it's 9 to 10. Okay.
I know 99%.
Okay, I need to refer to you. He asked some questions. And in the last minute. Okay. We are running this new process in the Faculty of Humanities.
Really?
Yeah.
Bringing exact science.
It's the
Bringing
second
the truth
year.
to.
It's really challenging.
I think that's fantastic.
It's a very interesting class. We have 100 students. It's amazing. So we teach, we call it neurodegenerative disease from research to hope.
Yes. Ah, very nice.
and we give them homework, which is also challenging. So they have to high power.
Yes?
Very interesting. Okay, and the leaves, because from Kazan there is nowhere, I'm not sure there is.
Don't worry, I will make it. It's just because I can be there at 9. So it's very important week. I need to. The traffic must be bad in the morning. I just think I need to drop the kids on the phone. Even should be fine. So you're using this slide using the ice model stuff?
No, we use mainly the neuromuscular junction.
I see. And you look at movement or
Much closer.
Yeah, but just as a model for neurotransmission. And so from there on, then you build on
what is the output you mentioned?
well, it's part frustrating who does the experiment, so but he has really a whole battery of analytes. So what he does is first electrophysiology, spontaneous activity, stimulated activity, high stimulated activities, then morphology, both electron and then SM143 uptake. And so then he has two protocols for reserve pool and for active pool. And what others, the morphology. For instance, we have an endocytic phenotype with LARC, and you have these satellite photons which nicely correlate with the endocytic phenotype. So it's, I'm really very excited about versatility. And then of course you have phenotypical changes like one knockoff slide, they have muscular degeneration of the thorax muscle, and then also five phenotype. So that's also used, but we basically use the neuromuscular junction to do genetics and find and so the funny thing is that these two, with pink, which is recessive, and black, which is dominant, in both cases we find neuromuscular junction phenotype. So if you do high stimulating activity, so high frequency stimulation, then you get a loss of the amplitude of the signal. And so, but the molecular cause of it is completely different. So in the black, we have shown that it's an endocytic phenotype, and in the pink, we have shown that it's a mitochondrial phenotype. But both finally give the same phenotype. So, and for LARC, it's very interesting because we could show that the phenotype is both gain and loss of function. So if you have overactivation of LARC or you block LARC, you get a similar phenotype. So which explains the mutations also, because we have one this famous A2019S, which is gain, but many of the other mutations, it's not clear what it does on kinase activity, and probably it's loss. So in the phenotype we have is loss and gain, because it's a phosphorylation cycle which is regulated. So LARC is responsible for the phosphorylation, and then there must be something which dephosphorilates. So if you have no LARC, you then get phosphorylation. And if you have too much LARC, you get too much phosphorylation. So that gives you twice the same endocytic phenotype.
You want some water?
I got some water. That's fine.
Or a coffee or something?
It's okay. I go on for another 45 minutes.
Yeah, we use it.
When we talk,
Like last note.
They courage.
Let's say take some questions and try to extract ideas.
Yeah, we probably do it tomorrow, but we express when it's dominant or RNAi only the peripheral neurons in the brain. And we just follow the behavior of the five because it's neurotransmitter cascade behavior.
Yes.
Yes, so we can really follow well, we have, especially with the polycule, that's the main setup for this. We can have phenotypes. probably very early. And you cannot really see a morphology from in the neurons, but you have a very
Functional.
functional phenotype. And this is very powerful to the experience because
Yeah. And the same with this neuromuscular junction. You don't need to see massive things. You have functional without.
And when we look in the mouse databases into protection and risk factors, we found a huge overlap. We looked at the data sets of expression in the brain of the different mouse models of Parkinson, and we found a great overlap of risk and protection factors.
Which are changed in, yeah, that's probably the reason why they have no phenotype in mice, or not so much phenotype, because they have a lot of protectors.
Yeah, mice are much better protected than we are. Cockroaches even better. The cockroach is even better protected. Oh yeah. When we'll all be gone, they are also protected against radiation.
Yeah, you can still take over.
When we lived in the Rockefeller University, you learn not less than a huge building with a full-time exterminator. He starts January 1st or whatever, after Christmas, in the first floor and goes up. And by the time he gets to the end of the building, he starts over again. And the cockroach is like the salesman from Hamlin. They go before him. It was terrible. We always learned about the American cockroach, and I only learned in New York how correct its name is. Okay, I think we need to call them back.
We have big baby who made this.
Yeah, brain center.
Yeah, you should do something in my department.
It works.
Yeah, it's
Send a message.
it shows what you want and it shows what you can.
The brain center people mostly electrostimulated. So if the senior scientist is there, they will stand it. Susan Bonini was a real problem. And I'm saying, hey, Susan, hey.
Yes, I can imagine. Yeah, I was a bit afraid for that also for tomorrow. I don't have electricity.
No, not having electricity is fine, but we need to avoid using our technical side.
Tomorrow it's more growth. No, this is more technical. Okay. So I hope you are ready for the next round of biochemistry. So please ask me questions, because I never know whether you understand, whether I go too slow, too fast, whether you want me to go a bit faster, you can say that, or slower, no problem. So this is work from Lucia, from Mexico. So I need to exercise to say her name in the right way. I have also Lucia from China. So and we say Lucia or Lucia in Dutch, so it's quite. And I can tell you Mexicans are very proud people, so you better speak correctly. So she's a great biochemist, and she's really focusing on purification of gamma secretase. She succeeded in getting a protocol where we have now milligrams of sustainable gamma secretase, which is active. So I think it's really a great achievement. And she also decided that it was important to really understand how mutations in percellin affect gamma secretase. So just to refresh, here you see a typical family tree of patients with autosomal dominant Alzheimer's disease. It's really, if you look to these family trees, you see how horrible or how terrible this is. So you have here somewhere grandparents. The father had Alzheimer's disease. Then two daughters. They both have Alzheimer's disease. They related to their sons in the next generation. And it goes on. Here you see a typical family. If they come, one of these patients come to the clinic, to the genetic clinic, the family tree is studied, and you see that brothers, sisters in two related families have 1 in 2 chance to get Alzheimer's disease. And so this is of course the generation of the truth. These are the kids. We don't know whether they will get Alzheimer's disease or not. And so, so we have mutations in preselin 1, mutations in preselin 2, a homolog of preselin 1, very similar, and then mutations in amyloid precursor protein. And what's also characteristic for these mutations is that the disease starts quite early, between 20 and 60 years old. So, while classical Alzheimer's disease is something from really old people, 80, 85 years old. So if you look to the mutations, I told you already about the measles in presenilin, but here you see a little bit more. They are in fact mostly in the transmembrane domains. And here you see the mutations of APP, so at the beta secretase site, a few in the site, and most at the gamma secretase site. So and again, you see mutations in the substrate of gamma secretase or in the catalytic subunit of gamma secretase are sufficient to cause Alzheimer's disease. These I skip, I've explained already enough. So what we know, and I will go into, what we know is that gamma secretase is this big bulk of proteins in the membrane, and it cleaves, a first cleavage occurs, and we call it the epsilon site, close to the cytoplasmic border of the membrane, and it relays APP intracellular domain, not intracellular domain, but I didn't tell to you that in the meantime there are at least 60 other substrates. Encadrin, neuregulin, LRP, name a couple of proteins, and there is a good chance that gamma secretase is able to cleave it in the membrane. So releasing by this epsilon, this first cleavage, the intracellular domain, which in case of Notch goes to the nucleus. For the other proteins, it's much less clear what the intracellular domain do. And then for APP, we know very well that there are a series of progressive cleavages which up to a certain moment release then the other side, which is the beta side. Also for the other substrates, there are several cleavages in the transmembrane domain, releasing finally the ectodomain. And so there is a big discussion always in science when something is invented, what is now the problem? I told you already my conviction that it's the gain of function or the change in function which affects a beta, which causes Alzheimer's disease. But there is also a whole school which say it's the loss of function, these mutations in the gamma secretase affect the release of intracellular domains, and it's the loss of function of this which causes Alzheimer's disease. And there is quite some discussion about it. What's the truth? So there has been these discussions, the loss of function mutations, when loss is a gain, blah blah blah. Well, our take was, from a genetic point of view, the mutations, because they are dominant, seem to result in a gain of toxic function. But from a biochemical point, we had already data that many of these mutations in gamma secretase affected the enzyme, and in my opinion, make the gamma secretase defective. The question is, of course, in what way defective? And there we see really the contrast between geneticists and biochemists. Geneticists talk about a mechanism when they have genetic interaction, when they can make arrows between different genes. A biochemist talks about the mechanism when he knows how two proteins interact with each other, change conformation, and affect things. And I think a lot of the discussion in the field has to do with talking different languages between these two fields.
But if you just put, they are beta peptides, you get toxicity also independently of a mutation in the enzyme that cuts it. Huh?
Yeah, of mutations in APP are sufficient to cause disease. Yeah, but anyhow, look, I have here a paper of Ji Shen. Ji Shen is quite proliferative in that field, but there are many other researchers also. And this was a comment she wrote when I think she succeeded in telling the opposite of what the genetics told us. So there have been a couple of publications where, for the first time, mutations were found also in the other subunits of gamma secretase. So the Alzheimer mutations are these missense mutations in presenilin. In these papers, they found mutations in nicastrin and in PEN2, which caused complete loss of functions. So there were not missense, but really loss of functions. So there were stop codons and frame shift mutations. And so by definition, in these patients you have only 50% genetically normal gamma secretase. So if it would be a loss of function disease, I would predict that in these patients you would have a very early onset Alzheimer's disease. But these patients present with acne inversa. That's a very severe form of acne, not the acne of the puberty, but really a toxic acne where you have and it continues over life. And we think, it's not proven, but I think that it has to do with decreased notch shingling in the skin. We know that notch is very important in the normal maintenance of the skin. And also, now I'm going to speculate. So what we know is there is a haploinsufficiency in these patients of gamma secretase, and we see the skin problem. What I think is that they acquire an additional mutation in notch at certain positions in their skin, and that causes locally this acne inversa. So this has to be further investigated. But the main point I want to make is that in these patients there is no, at least not reported at this moment, an increased incidence of Alzheimer's disease. I don't know how G came to the conclusion that this fitted in her hypothesis that loss of function of gamma secretase causes Alzheimer's disease. But in any event, I'm going to skip this slide. I made a discussion. In any event, the conclusion Lucia and me made was that if we want to really finish the discussion, we need to understand what the mutations are doing biochemically. And so we set out. So if gamma secretase loss of function is sufficient to cause FID, we should see only effects at the kinetic properties of the enzyme, and ABP should affect also the kinetic efficiency. If it's a dysfunction of the gamma secretase, we would see changes in A beta production without necessarily changes in the release of the intracellular domain. So to investigate this, you need in vitro assays, and so there are three important questions which you have to address if you want to understand the biochemistry of gamma secretase. That's first, how the substrate gets docked to the enzyme. Second, how the first cleavage occurs, and that's very interesting. You're probably not biochemists, but you will immediately understand the point. So the first cleavage occurs in the peptide. So it's an endopeptidase-like cleavage. The next cleavages, which cuts further, are in nature, biochemically in nature, different. They are a kind of carboxypeptidase activity. So that's also the reason why we discriminate between the epsilon cleavage, which releases the intracellular domain, and the carboxypeptidase activity, which releases the peptides. Is that clear for you? So in here, then the mechanism which we, the way we think that it happens. So you have first this epsilon cleavage, and then you have progressive cutting. And what you see is that this transmembrane domain of amyloid precursor protein, in this case, becomes shorter and shorter. So at a certain moment, it's not hydrophobic anymore, and it can be released into the extracellular medium. So that's the way we think. And then very nice work from Ihara, from a Japanese group, has shown in vitro that if you look to these progressive cuttings, you can basically define two cleavage pathways. So you have ABP, which is then first cleaved here at the border by epsilon, but this can occur at two different sites, two amino acids different, so you get a cleavage between amino acid 50 and 49, or between amino acid 49 and 48. Is that clear? And then you get progressive cleavages every three amino acids. Why this is every three amino acids, we don't know. It could have to do with the alpha helix, but we don't know why this happens. But Ihara has shown in vitro that you can find the three peptides between every cut. So I think that's a very good proof that this mechanism occurs. So you have then A beta 49, A beta 46, A beta 43, A beta 40, and this is evidence that A beta 37 can be generated. And in the second production line, you get A beta 48, A beta 45, A beta 42, and A beta 38. And so what you usually find in the literature is that A beta 40 and A beta 42 are the important peptides, and that in the genetic mutants, A beta 42 increases and A beta 40 decreases. nobody understands. Well, one of the possibilities, of course, that in the mutations you shift into this pathway. Okay, I'm coming to that. So this model was used to identify, to clarify what happens with 6 different mutations over the precedent. So we took 6 different mutations at different positions, thinking that they would have possibly different effects. Here you see the protein expression in the membranes we use, so you see it's very, very equal. So we have equal concentration of enzyme, and we added amyloid precursor protein, but also Notch, because loss of Notch could be involved in Alzheimer's disease, and 2 other substrates, UP4 and cadherin. So we added in cell-free experiments, and here you see the complicated, we did, Lucia did many, I think hundreds of these Western blots, to carefully quantify the reactions, controls with inhibitor, and then here you see the generation of this intracellular domain. And since we have now a system in solution, we can do kinetic analysis. And don't worry too much, I'm going immediately to the conclusion here. But what you can do in these experiments is you have solubilized enzyme, you have solubilized substrate, and so you can increase the substrate concentration, which you cannot do in cell-based assays. And so then you can make these kinetic curves, and you probably remember from your course, your basic course, this has Michaelis-Menten kinetics, and you can start to determine the max and Kcat and Kgs and Kms, and you can basically measure the enzymatic efficiency of the different enzymes. And here you see that these curves are quite different. Here you see the curve for C99. This is the wild type curve. These are the 6 different mutants. This is the Notch curve. You see also again different mutants here for L4, etc., etc. And so if you measure the kinetic efficiency, which is the most important catalytic parameter of the enzymes, you see here the different results. For wild type, we put it at 100%. You see that in several mutations, you get indeed a loss of catalytic efficiency. But you see also for this mutation, for instance, that there is no effect in the catalytic efficiency. So this mutant processes both APP and Notch to the same extent as the wild type. So you don't get Alzheimer with this mutation because of a lack of catalytic efficiency of your enzyme. And also here you see, for instance, these 2 mutations don't affect Notch.
What about protein-protein interaction?
What do you mean?
Would this mutation change the way this variant interacts with other
With the
proteins?
substrates?
Yeah.
Well, we don't think so, because this is a cell-free assay where you have the 4 proteins together. So if they would affect it, you would have a lot of branches. And second, we don't see an effect. With the same concentration, you have the same product. So that tells you also that the docking of the substrate is similar in this complex and in this complex. But I come back to the mechanism behind this loss of function in a minute. And so then we checked also for other substrates, and here you see again M139V. It's a bit kind of special because N-cadherin seems to be even better processed by this enzyme. But for instance, ERK4 is not affected. And also with other mutations, we do not see directly an effect on other substrates. So it's certainly not the case, and I think that's the main conclusion, that the reduced release of intracellular domains is the common cause of these mutations. So then what would be the common defect in all these different mutations? So we went on for the next cleavages here, these different peptides, and so we have not ELISAs for all the different peptides. It's actually very difficult to make antibodies, specific antibodies against these hydrophobic ends. I would love to have them, but we don't have them at the moment. So we use the real gels to look for the A-beta profiles. You see here the very different A-beta profiles. You see here that M139V. No, let's go to the wild type. What you immediately see is that several of those mutations, including M139V, have accumulation of longer A-beta peptides. So it seems that the turnover of these longer peptides is somewhat affected. And to do this quantitatively, we used ELISAs for one of the cycles. So you have a first cycle in the processing, a second, and here you have this third cycle, or fourth cycle if you start this one. Let's talk about the fourth cycle. So you have the first, the epsilon cleavage, then 1, 2, and 3. So you have the fourth cycle of the processing. And so the interesting thing is that this process A-beta 43 to A-beta 40, and A-beta 42 to A-beta 38. And so here you see we set this at 100% for then we compared the different mutations at this level. And so the first thing you see immediately is that in all the mutations, this doesn't work. So in all the mutations, the A beta 40 and A beta 38 is lower. Here, 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 and here. So this is a common phenomenon in all these mutations. And what you also see is that the longer A beta peptides are increased relatively. But what you also see is that, for instance, this mutation gives you in total less A beta. So there's also a loss at the level of A beta production. So you have a mutation which doesn't increase the A beta levels, which increases the amount of A beta 43 and A beta 42, and decreases the levels of A40 and 38. So in other mutants, you see an overall increase of A beta production as well. So this, what do we learn from this? Well, what we learn is that I'm looking for the next slide probably. Yeah, in the next slide, what we did is actually we put A beta 42. We made instead of the A beta 42 to A beta 40 ratio, which is used in most of the clinical studies, we made the ratio of A beta 43 to A beta 40 and A beta 42 to A beta 38, which makes much more sense because that's in fact showing you the efficiency of the enzyme. And then what you see here with all the mutations is that they in fact, both for the two ratios, they affect the fourth cycle of the enzyme. And so how can you mechanistically interpret it? Well, it's in fact very straightforward. It means that the substrate, when it's bound in this enzyme, is released more quickly than in the wild type formation. And that makes perfectly sense. So the mutations affect the time that you can keep 43 or 42 in the enzyme. And that explains, first of all, this change in ratio 43 to 40 and 42 to 38. But it also explains the increase in beta 42 to A beta 40, which has been for a long time seen as the signature for Alzheimer's disease. So the conclusion from this story, and I'm going to skip the PS2 mutation because they basically confirm what I told you here, that the crystalline mutations, both in PS1 and PS2, cause a qualitative change in A beta, not an increase in A beta, but a qualitative change in the beta profiles. So this is a PS2.
Sorry.
Yes.
What about the folding of the peptides? Could be that the different efficiency in cutting is related with the more or less time to fold differently and.
Yeah, well, I think basically that the more rapid release of these more hydrophobic peptides cause folding problems in the extracellular medium. And so what we learned from this also is that not only A beta 42 is a problem, but also A beta 43, which has now a couple of months ago been stressed also in publications. So, and I think that the entire focus of the field on this A beta 42 and A beta 40 is not correct. We should broaden our perspective. It's a qualitative change in the A beta profiles, which is probably this A beta sub, which I called. The clue to understanding the toxic species has to do with the biophysical properties of this mixture. So that's what we conclude here. PS1 mutations cause AD by qualitative and not quantitative shifts in A beta. Yes?
Most of the cases are sporadic.
Yes, that's a good question.
So, I mean, do you have any idea what happens in sporadic cases? I mean, if a person is changing the activity, something.
Well, for the sporadic cases, I think that the best ApoE4 story is the best story there. So for ApoE4, I think we have two mechanisms. One is that it affects the clearance of the beta peptides, and one is that it changes the blood-brain barrier, which is a completely different story. But let's focus. So what I think is sporadic is that it has to do with clearance, and with differential clearance of the more soluble forms versus the more hydrophobic forms. But we have also just published a paper where we show that nitrosylation of gamma secretase, which occurs when you keep, for instance, neuronal cultures for a while, that it affects the enzymatic activity and also the ratio. But I don't think that, I think that most of the sporadic cases will be explained by clearance mechanisms. But the main thing is that that it tells you also that increased A-beta and getting and lowering it, that we should not be obsessed with lowering A-beta, we should be more obsessed by changing the patterns of A-beta. That I think is the most important lesson for sporadic Alzheimer's disease. Just kidding, because I'm going to skip a couple of discuss. So then we went also to the APP mutations, so the substrate mutations. Do they affect this type of cycling? And so again, to make a a long story short, here you see this A-beta 4340 and 4238 ratios. They are not affected by these APP mutations. But what we see there is, if we make now the 4342 ratio to see what production lines are affected, you see that there's a clear decrease of this production line, so 4342 or 4038 if you make those ratios, and an increase in this production line. So the question is, what happens there? Well, what we think is that the substrate, because these mutations are all in the gamma secretase site, that the substrate binds slightly differently in the binding pocket versus the wild type. And so to check whether it is indeed the first cleavage site which is affected, we did the following experiment. So we got specific antibody which recognizes the intracellular domain cleaved at the 49 site, or the intracellular domain cleaved at the 50 site. And so this neo antibody recognizes this, gives you a red signal, and when you use the green, the flag tag, you see the two bands, and that's given here. And so we can then do the overlap, and you see that we have different colors of yellow and green, depending on how much the cleavage occurs at this site or at this site. And so if you look here to the APP mutations here, what you can see is that these 4 mutants all decrease the red signal. So they shift indeed at this site the processing to this production line. So with the prisoner mutations, we have also some cases, actually 4 out of 6, which does the same. So you have there 2 mechanisms in the prisoner mutations. First, the initial docking is slightly different, and second, the 4th cycle is different. So is it clear for everybody how these APP mutations affect the ratio? Because they will drive, of course, more substrate into the 42 pathway, and less there, which also would cause a relative change. And in principle, it would give you the same problem. So our results rule out the possibility that loss of intracellular signaling is necessary and sufficient to cause Alzheimer's disease, as postulated by simple loss of function hypothesis. I'm stressing this because it has been used as an argument against gamma secretase inhibitors, because they would mimic this loss of function, but I don't think that that's the real problem. And conclusion 2, I've stressed already to you, so you have 3 mechanisms, if you want, which cause the mutations. So, prisoner mutations purport the more rapid release of the hydrophobic beta peptides. APP mutations shift the processing into that pathway, and in some prisoner mutations you have a combination of the 2 mutations. And this can explain, if you go into the literature, a lot of data which have been published, for instance, why certain gamma secretase modulators affect prisoner mutations or not, etc., etc. But I have no time to discuss those things. So the qualitative shift in the data profiles is the common denominator. And maybe I'm going to finish with well, I have still some time, okay. So, yeah. So I hope it's clear to you how these clinical mutations affect personnel and APP mutations. No questions? Then I'm also not going to ask questions. But so I'm stressing the importance of these A-beta profiles. And so we started to do some experiments. You know that I don't like this oligomer and A-beta sub hypothesis because the definitions, it's not that I don't believe the hypothesis, but it's because I think that the definitions are not stringent and not clear. And so one of the experiments we wanted to do at a certain moment is, if this A-beta sub hypothesis is right, then small changes in the A-beta concentrations will affect the behavior of the A-beta peptides. And so that was the question we wanted to address in this experiment. It has taken us for years to get this really working and to get this really reproducible working. So what we basically did is we changed the relative concentration of a long A-beta 40, and a short A-beta 40. A-beta 42 is a long, and A-beta 40 as a short. It's an in vitro, because I think if you want to mimic the in vivo situation, you need to use A-beta 38, 43, etc., but then it becomes an impossible experiment. But we decided to use this and to use two ratios. So we used a mix in which we have a 1:9 ratio 42 to 40 and a mix of 3:7 ratio, which is really a small difference if you think about it. And we postulated that this change would affect biophysical properties and biological properties. And I'm going to summarize those data, which were produced by Irina and Faina. Faina is Israeli and Irina is from the Ukraine. And they have worked beautifully together. And we have also Lebanese people in the lab, and they also are collaborating. So science is really, making the world one community. It's one of the things I love. So the first thing is proof that we made the right mixes. Here you see mass spec. So if you have the 1:9, you have this big peak here of I think it's of 40, and here small peak of 42. And if you have the 3:7 mixture, you have this peak of 40 and peak of 42. And the first thing we did was biophysical properties of these mixtures, so you can follow the polymerization of these peptides over time. And you see, just focus on the 3:7 ratio. You see that this has the tendency to fibrilize quite rapidly. And then you have a very slow process, well, a rather slow process, until it becomes completely fibrous, and you can measure that with the alpha-V incorporations. With the 1:9 ratio, you see that there is a long phase before the oligomerization starts. And once the oligomerization starts, or the beta sheet formation starts, it goes much faster than this mix to reach the plateau. So that's already a first demonstration that the ratio of these peptides is very important. And here you see electron microscopy of the fibril formation. And again, let's focus on those two. You see with the 1:9, already rather quickly, these fibrils, these mannose oligomers, and if you add that to a cellular culture, nothing happens anymore. And you see that with the 3:7, it takes some time. You get these ugly intermediary structures, and then after 10 hours, they start also to form this A-beta sheet. So you see a small shift in A-beta 42 to A-beta 40 has dramatic effects on the biophysical properties. So unfortunately, we don't know what happens between this monomer and this fibril formation. We don't know, we have no tools to see what happens at that time, but we can measure what happens at that time with assays. And here we used microelectrode array, probably you know better than me what it does and what you can do electrophysiologically, but we can use this to culture primary neuronal cultures, and then to add our A-beta mixes, and we can have a very rapid feedback. Between 20 and 40 minutes, we see changes in the spontaneous electrical activity of this neural network. And so we thought this is a good way to measure very acutely, with hopefully little changes in our mixtures, on the activity. And here you see a relevant profile. You see, for instance, here when we have the control, you have nice spikes, 10 minutes, 20 minutes, 40 minutes. A-beta 40, in fact, it increases the frequency of the activity. So even A-beta 40 has effects at the level of the synapses. With the 1:9, we don't see too much of differences. It looks okay. But with the 3:7, you see really a decrease in amplitude and also a decrease in frequency. So this 3:7 has, at 2 hours after preparation, a quite toxic activity in our neuronal culture. If we wait for 10 hours, this toxicity is gone. So the conclusion from these things is that it really fits with the hypothesis we made at a biochemical level. So the mutations change the A-beta soup you generate. Small changes in the mixture of the soup change significantly the biophysical properties and also the biological properties of the A-beta mixtures. So I would like to wrap up the gamma secretase and the A-beta with one last thought. So maybe I should first conclude from a practical point of view. So what have we learned from a practical point of view with things I was talking to you about? So first of all, I think that gamma secretase remains a very interesting drug target. But we should look at it first from a biological level. And so what we should avoid by all means is to affect biological relevant epsilon cleavages, which gives you these intracellular domains, especially much. Because if you affect that, if you don't find the therapeutic window around that, you will have side effects. But this is not different from any other drug. There are basically every drug who works has side effects. So I think we need to work much harder to find the therapeutic window. Second, the A-beta profiles are important. So we know that the clinical mutations cause lower A-betas, not only 42 but also 43. And what I didn't explain to you, but with some of these mutations we are suspicious that part of the A-beta sits at this level. So lower A-beta are not very good. And so I think that the gamma secretase modulators which are currently developed to increase the production of shorter A-beta peptides are a very good path to go. And secondly, something which is not considered too much, but we know from the APP mutations that the APP mutations drive APP into this production line. And so many of the gamma secretase modulators are optimized by increasing the A-beta 38. But I think it's dangerous because you would select for modulators which increase this pathway. And I think we should look for modulators which increase this pathway and even go to A-beta 40 or even go to A-beta 37. So that's the second important.
Which is the opposite.
Which is in fact the opposite. But we have tested a couple of gamma secretase modulators in the lab, and most of the modulators increase this at the 2 levels. So it's not too bad news for the drug development at the moment.
It's coming back to like maybe a few slides before, but because it's the ratio what is important. I guess it's very easy to test even in vivo by generating a APP which can give only one of the products. The one
that would be a beautiful experiment to do, but we don't have such mutants. So if you make a mutant in the APP, you change the profiles, but you get still a couple of other peptides. We have injected those beta 40 and 42 mixtures and we see acute neurotoxicity in vivo. So it translates in vivo, but it's very difficult to do that in a controlled way. But it would be a very good experiment. I agree. I think Todd Goldie has done some experiments showing that if you overexpress only a beta 40, you don't see toxicity.
But you don't even need the context of the secretase and all this. You could even put like a step protease or whatever is cutting and releasing a peptide.
Yes.
Like this to see if you can rescue this balanced ratio.
I think it's a good experiment to do. We are not doing it, but it's really something which we should keep in our minds. Yeah. So a second point I would like to make is when to treat, because what is clear, I think, from what I've explained to you is that a beta is a driving force in the disease. But what is not clear to me and to anybody in the world is whether when the disease is fully blown, when your brain is full with a beta, when you lose neurons, that it's still a good idea to try to lower a beta at that time. It's like a heart infarct. You have a lot of cholesterol, your arteries get clogged, you get a heart infarct, your heart part of your heart dies. Well, at that moment, treating with cholesterol and lowering your plaques will not do much anymore. And I think with Alzheimer, we are coming into a similar line of thinking. Because one of the reasons is that we know that this line presents, this is time, and this red is the increase of a beta in the brain. And as you see, deliberately we draw this line already starting maybe in the 40s, but certainly in the 50s, people start to accumulate already a beta. And we think that some of them, many of them actually, will go further into full-blown Alzheimer's disease. So So you have this early accumulation, and then in the late, well, 60s probably a bit too pessimistic, but you see, well, in the 60s you start probably to have already a lot of amyloid in your brain, and here this blue line represents, in a simplistic image, accumulation of tau, and what we think, and there is good reason also with the Bragg staging, the accumulation of tau reflects much better the progress of the disease, of the disorder, and is much closer associated with the disease. And what you see here is 3 levels of a beta. So L1, L2, level 2, level 3. And so what we are thinking is, if a beta is a trigger, so you get a beta, and it starts at a certain moment to trigger also tau formation. Then if you can lower a beta before it has triggered the tau levels at a certain level, you will prevent Alzheimer's disease. But if it's a trigger and you would start to lower a beta even at this time, when there is already tau pathology, you would not do anything anymore, because it's not necessary anymore to drive the disease. Of course, if a beta is a driver of the disease, like it's imaged here, and you would lower a beta, then you would lower tau again, and you would cure the disease. So the truth is probably between the two. I think more and more, and my colleagues too, think more and more that a beta is likely more a trigger. So you need to treat before you get already the tau pathology running. Maybe it's a threshold, that would be also fine, so that you need to reach, for instance, level 2 to start the tau pathology, or to get tau pathology independent of a beta. If it's a trigger, then it would be still okay to treat at that time. But what this scheme tells you is that it's probably a desperate attempt to lower a beta in already clinically manifesting Alzheimer's disease cases. And all clinical trials at this moment occur at that level. So what we need is a quite safe anti-gamma secretase or beta secretase or an antibody which we can use in patients which are at the level of triggering or even in the preventive at the preventive level of disease. So is that clear for everybody? Because it's very important for the next years to understand the discussions about drug trials, why drug trials are failed. You will see a lot of pessimists who say anti-amyloid, this proves that the amyloid hypothesis is not right because it didn't work. And you will see then optimists like me who think, okay, we are learning, it's probably we need to develop a preventive therapy, and the decision finally is made by the people who have the money. And so they don't do the research, so the researchers who shout most loudly will convince the companies to spend another $600 million on a next experiment to see whether we can prevent Alzheimer's disease or not. So I think this discussion is really not only science and insight, but also really deciding about the big politics in Alzheimer's disease. So, well, I see I still have in principle half an hour, but I will shorten it to 15 minutes to talk a little bit about another favorite drug target, the beta secretase, which cleaves at the amino terminus of a beta, and which is a much easier drug target. If you take base 1 knockout, the mouse look not normal, because that's really very exaggerated. No gene, a knockout of a gene will give you always a phenotype, but they look much better than any gamma secretase knockouts which have made. If you do interference of base, you can indeed prevent amyloid pathology in transgenic mice, so you need only a 50% decrease in base activity to get a significant effect on amyloid accumulation. And the third, we have no genetics involving base, the beta secretase in Alzheimer's disease, but we have good evidence that base is elevated in a subgroup of patients, which makes it logical to decrease base activity. So it's a very simple enzyme. It's a membrane-bound enzyme with an aspartic catalytic domain. There are crystals. But the big problem is that inhibitors, which have been made over the last years, are all susceptible to PGP, P-glycoprotein efflux from the brain. So you make an inhibitor to base, a very specific one. You give it to a mouse, a rat, a human. It goes faster out of the brain, then it's transported into the brain. And that has been really a big problem. And the other problem is the specificity, but that has been less of a problem. So you have a couple of enzymes which are related to base 1 and base 2. And so for a while it was difficult to target specifically base, but that has been solved. So when we were reading that literature, we thought, why, if you can use immunization to tackle A beta, so you can get antibodies into the brain, maybe we can make also very selective monoclonal antibody for base and target it. And maybe by doing that, we will not only target the catalytic site of base, but maybe we find also sites which are involved in the regulation of base. Exosites, how do we call it? Allosteric sites, which you can modulate and which you can use for development. And I'm going very quickly through this data because it's, you can find it in the publication. It's very technically. So we made monoclonal antibodies. We used knockout animals, which gives you a very good way to make very specific antibodies. And so we finished with, and we ended with a very nice antibody, which is effective in the nanomolar range, lower than nanomolar range, and which blocks completely A beta in primary neurons. So we were very excited, and so we tried for 2 years to get this monoclonal antibody in sufficient concentrations over the blood-brain barrier in mouse. And then we became nervous because we heard from Genentech that they had developed a similar antibody, and that they had a chimeric antibody with transferrin receptor, and that they were able to get it over the blood-brain barrier in monkeys. And so we said, okay, we are going to do the shortcut. We inject the monoclonal antibody in the brain of mouse, and we could show indeed a significant, or no, but a significant effect on A beta processing in the mouse brain. So we were quite certain that this was a good monoclonal antibody, and that basically we had to transfer it to a group who was able to transfer the monoclonal antibody over the blood-brain barrier. So yeah, we are working with a couple of groups to do that. But in the meantime, we try to identify the spot, the site where this monoclonal antibody binds. And here again, you see we did a lot of experiments, first with truncating base, seeing where the antibody bound. And then at the end, we did site-directed mutagenesis. And we could show that the base antibody doesn't target the catalytic site of base, but here is 2 loop domains at the outside of the base protein. And so that's very interesting because this is a complete unique structure. You don't find these loops on base 2 or on any other protease. So we knew that this was really a very specific inhibitor for the base protein. And the question was, how does this block base activity? And so the most annoying or boring explanation would be that it just sterically hinders the substrate. But I think there is a much more interesting explanation, because this loop domain, especially F, is in competition with an alpha helix, which regulates the binding, one of the binding sites, the S3 subpocket, for the substrate. And so what we think is that our monoclonal antibody fixes base in a conformation which doesn't allow anymore the substrate to fit in that subpocket. I don't have the final proof, unfortunately, because, well, we are working together with the group to co-crystallize the antibody with this, but we feel strengthened in this hypothesis because another pharmaceutical company did screens with peptides blocking base, and a couple of their peptides, and they have crystals, bind exactly this site and blocks also the processing of base substrate, but at a micromolar concentrations. So this fits with the hypothesis, and we are now trying together with the company to make small molecules targeting that site, and we are also trying to make our monoclonal antibody passing the blood-brain barrier, which is one of the most interesting things. And then the last proof that this site is important was site-directed mutagenesis, so we make a few amino acid residues in this loop F or helix A mutated, and you see we get nice expression, and you see that in the mutation in loop F lowers the activity, and the mutation in helix A increases the activity, further confirming that we found an allosteric site in base important for drug development. So, and I think that I will skip here. We have but you can find this publication if you are interested. So base, the link between base activity activity and regulation by microRNA network. But this would take me too much time, I think, to introduce the microRNAs, etc. So I'd like to thank you for your attention. If you have further questions, I'm very happy to try to answer them. Some of you I will see in the next days, so I look forward to your interesting slides. Thank you for your interest and attention.
You showed us an experiment where you took an electrode and put your arm in these neurons and then the A beta. So did you look at like transcription or proteins that are going up or down? I mean, looking
No,
at
we
what
didn't.
happens.
David, I think this is an excellent proposal, especially because with A beta 40, you see this change in electrical activity. I think that these effects we show are acute, so I do not expect that they are directly affecting gene transcription, etc. But it could be that further downstream that they change. Yeah, it's a good suggestion.
If not, then thank you again. And remember everyone, hello, lecture tomorrow, and the seminar on
So I think that while you would affect upstream also, you would also affect a couple of other physiological processes. So I don't think it's a very good experiment. You do a lot of
So we will give you a new paper. Yeah. And they will show
gamma mediated.
molecular and
One
one
molecule
No.
is here and the other one is here.
No, so that could also be that you would see also random agents. So that's no risk first. And then we have hypothesis and mutation in your sequence. And then you can block formation of more downstream.
You showed.
Okay? And you
Well, no,
have
I
to
didn't
write a
mention
short
that
essay about
we will, we have
Okay, when do we start?
a detailed experiment.
we'll put it on the website and we'll let you know.
Okay.
And then you have,
Okay.
okay?
Yes.
These are biochemical events happen throughout the life of the individual. While they are more, while the age is more susceptible to accumulation of
Well, there is two separate. I think that in aging you get a decrease of the clearance. And then we have also data now, and we are going to work further on that, like nitrosylation of the gamma secretase affects the profile in a similar way as the clinical relationship.
the only uneasiness I feel about this, I'm fine with the inverse direction regarding A beta. You presented that this is a single sequence of events, and of course
Yeah, I know.
it talks to other.
I know.
I'm cholinergic.
Yeah, I know. But I think that the cholinergic is not what you will be delighted. The most changed microRNA in our brain is 120. Yeah. It's incredible. Yeah. So, but yeah, of course, I mean, any aging disease needs a factor.
And it's not only cholinergic and it only starts that way, but I don't know yet which is the issue.
I think that that's something where I think it's well, there are other things which can converge on it. So you can have other problems like this clearance, there is the enzymatic effect. This may be physiological events or pathophysiological events which make you getting more A-beta. But I think that A-beta is a necessary part of the
Yeah,
cascade.
I have no doubt about it. The question is, which is the chicken, which is the egg? Do you use cholinergic neural stage and that triggers A-beta, or is it the other way around? I don't know yet.
Well, you
People
will never
ask
know.
me.
It's just,
That's why I'm
well,
asking you.
I think that you need to do these experiments, or you need to have patient data. The problem with patient data is that the only real experiments you have are experiments from nature, and are the clinical mutations. And there,
But that's a
very
different story altogether. That's exactly what I'm.
I don't know.
I mean, it gives
But
very
it could
important
be.
information,
Yeah.
but it is not necessarily the same type of disease. That's one equation.
But look, if you have familial hypercholesterolemia because of your LDL receptor,
Yeah.
you get high cholesterol, you get cardiovascular disease, blah blah blah.
Yeah.
There are 1,000 other reasons why you get heart infarction, and there are even a couple of examples of patients which have not a high cholesterol and still get heart infarction.
Right.
But what I think is that in the big bulk of cardiovascular disorders, cholesterol is a very major risk factor and a driver of the disease.
Yeah.
So that's my idea about A-beta too. So there are probably many reasons why A-beta can increase. I'm certainly ready to accept that not all neurodegeneration, I would be foolish to claim that it's driven by A-beta. But I think that a large part of what we diagnose as Alzheimer's disease has as a major element, just as a major risk element, A-beta. That's my opinion.
That I have no
Okay.
problem with.
But I agree that it would be very narrow-minded to reduce it to A-beta and so. That's
And then fight each other.
That's ridiculous. That's
Oh,
ridiculous.
stupid.
That's ridiculous. No, that's ridiculous.
I think we need to disconnect the projector. And would you like to take Bob down to the third floor, the taxi will be there?
No problem at all. Yeah.
Perfect.
Question.
And you know that tomorrow the lecture was moved to 5. Do you?
No, that's they have it at 5.
No, yes.
It's right.
It is probably.
But they move. I
It
think they
was
move.
2 o'clock and they switched
No,
it.
they
No,
move
it's
over
fine.
lunch,
I know
I
it's
think,
fine. It's at
Yeah,
5.
you know it's fine.
Yes,
Okay.
and
But nothing else was moved, eh?
the other is on.
Yeah, there is
that's
also
on
5.
5.
Yeah.
Ah, okay.
But
That
they
is
move
always
something else. I think they emailed me. They
Yeah,
move-
tomorrow well, yeah, but that's also indicated.
I
We
think
are
they moved the
making
lunch.
you work.
But they are going to
very
lunch with me. I think they move it. 1:30. I think they move it. But doesn't.
That I will see.
Yeah.
Tomorrow I have to be here at the
Yeah.
time.
Yeah,
Can I
we-
take that because as a
Sure. This is
Doesn't-
yours.
Yeah. They move. I think they move someone up.
That's fine, there's no problem.
You will shut the
Very
starting at-
dynamic secretary.
No, shut up. It will be.
She takes initiative, so.
And I need to
Anyway.
be in charge of this. Otherwise she.
Tomorrow it will be more general the talk.
Yeah. Yeah. And don't be surprised if someone will ask you questions like
Silly question.
That's not. There are no silly
I
questions.
think, well, excuse me, what is RNA? I
Oh,
had
really?
that one, not in this kind of lecture, but we have the computer students who go to biology lectures. And
It is more of their problem that
sure, but it's the lecturer's problem too. These are bright people who decided that biology you can learn by reading. So they come to the lesson and then they realize that they don't know the very basics. That happens.
Okay, you have everything?
I have everything, yes.
I think the worst was when I once said, if I use technical jargon, please let me know. So someone raised their hand and said, excuse me, what is jargon?
Yeah.